Assalamualaikum everyone, welcome back to this channel. So this is me Arya Wahid, and in this video we are going to start a new chapter of microbiology, and this is about the industrial microbiology. So let's start our lecture. What is industrial microbiology? It is a branch of science in which we are studying, or we deal with the products which are produced on large scale with the use of microorganisms. So if you are producing the industrial product with the use of microorganism, then it is will called a industrial microbiology, meaning the use of microbes in the industry to produce some products on the large scale. For example, alcoholic fermentation is done by this method, and we will produce alcohol by this process. So there are some properties of microorganisms which are able to produce a large quantity of things, like the microbes which produce spores, which can easily inoculate, which grow rapidly, which help to reproduce more products quickly, which should not be pathogenic, should reproduce rapidly. Some industrial products. So now we have discussed the properties that they should be easily inoculated, grow rapidly. Should produce the microbe or the product rapidly, and they help to produce the product more quickly, fastly produce it, and also it should not be pathogenic, not disease causing, and should reproduce itself rapidly. You should increase its yield. So some industrial products are also given here, which are helping to produce the microbes, like these products which are produced as a result of this process. This is called enzymes, antibiotics, steroids, alkali, food additives, various chemical like ethanol, citric acid. All of these chemicals are produced as a result of it. Now we are discussing some types of industrial product. First is the primary metabolite and secondary metabolite. Primary metabolite, these are the products which are produced during the exponential growth of that microorganism, like alcohol, and some are secondary metabolite which produced during the stationary phase, like penicillin. Now let's discuss them in the next page. So here I have drawn a graph between the time and between the level of sugar, alcohol, and cells. So this is representing the primary metabolite, and this is representing the secondary metabolite. So here you can say, as the concentration of sugar decreases, the concentration of microbial cell and the alcohol is increased. It means the cells are using the sugar, and during their exponential growth of that cell, the product, which is the alcohol, it also produced itself. But if you see in the secondary metabolite, you see the sugar is decreasing and the cell number is increasing. But the penicillin produced at this time. Here the alcohol produced at zero time, okay, or at one time. But in this process, the penicillin is produced after some time. So when the growth of cell becomes stationary, then the penicillin growth starts itself. So actually, the molecules which are produced by this secondary metabolites, they are the large organic molecules, require large number of enzymes for every steps. Like tetracycline is antibiotic, it requires 72 enzymatic steps. इसका मतलब क्या है कि tetracycline 72 steps में प्रोड्यूस होता है, और उसको 7 हर स्टेप में enzymes चाहिए होते हैं. तो हर 72 स्टेप में इसको 72 enzymes चाहिए हैं. सो ये चीज़ आ जाती है सेकेंडरी मेटाबोलाइट्स की, कि जिनको प्रोड्यूस करने में इतना ज़्यादा टाइम लगता है, स्टेप्स होते हैं, जबकि प्राइमरी मेटाबोलाइट जल्दी से प्रोड्यूस हो जाता है. अब हम बात कर देते हैं फर्मेंटेशन की. तो मैंने आपको बताया था, जैसे इंडस्ट्रियल माइक्रोबायोलॉजी की एग्जांपल में अल्कोहलिक फर्मेंटेशन में बताया था, तो फर्मेंटेशन को अब हम डिस्कस करने वाले हैं. सो फर्मेंटेशन है क्या? से एक्चुअली इट इज़ अ केमिकल ब्रेक डाउन ऑफ अ सब्सटेंस बाय द माइक्रोऑर्गेनिजम्स, कि आपके माइक्रोऑर्गेनिज्म एक सब्सटेंस को केमिकली ब्रेक डाउन करें और उसकी वजह से एफरवेसेंस और हीट रिलीज हो. इस प्रोसेस को हम फर्मेंटेशन कहते हैं. फर्मेंटेशन की फर्दर दो टाइप्स हैं, एरोबिक और एनारोबिक. एरोबिक फर्मेंटेशन की बात करें तो इट इज़ द सेट ऑफ केमिकल रिएक्शंस इन्वॉल्विंग इन द प्रोडक्शन ऑफ एनर्जी बाय द ऑक्सीडेशन ऑफ फ्लूइड इन द प्रेजेंस ऑफ ऑक्सीजन. क्योंकि ऑक्सीजन की प्रेजेंस में ये केमिकल रिएक्शन हो रहा है, ब्रेक डाउन हो रही है, एक सब्सटेंस की एनर्जी निकल रही है, तो इसको हम एरोबिक फर्मेंटेशन कहेंगे. मोस्टली साइटोप्लाज्म और माइटोकॉन्ड्रिया के अंदर यही प्रोसेस हो रहा होता है. और इंडस्ट्री में भी अगर हम ऑक्सीजन की प्रेजेंस में इन केमिकल रिएक्शंस को करते हैं, तो हम इसको एरोबिक फर्मेंटेशन कहेंगे. और इससे क्या क्या प्रोड्यूस होता है वो मैं आपको नेक्स्ट स्लाइड में दिखाती हूँ. Let's discuss the examples of aerobic fermentation. So various kind of antibiotics, enzyme and vitamins are produced by this process. Let's discuss the anaerobic fermentation. So it is the chemical breakdown of organic substance into ethanol and lactic acid. So in the absence of oxygen, so you are doing this process. So lactic acid, ethanol, and wine is produced by this process. Where various types of fermentations are given, like two processes involved in it: batch fermentation and continuous fermentation. So batch, what is the batch fermentation? So in this method, the nutrients are added in the fermenter. So first of all, like suppose this is a fermenter, you will add nutrient units, and after several days, the product will form it, and then we will extract out the product, and the tank will clean again and prepare for the new batch. So after several days, you will extract the product, you will clean the fermentation, and again you will add your nutrients to continue this fermentation process to produce a large scale product. Now we have discussed the batch fermentation. Now let's discuss the continuous fermentation. So let's discuss about the continuous fermentation. So continuous fermentation, its name indicated that continuously a process is occurring. So the addition of nutrients and the removal of product occur at the same time. So at the same time, you will add the nutrient, extract the product out. So this process will continue itself. So now I have also draw a diagram over here. So what is a fermenter? It is an apparatus that maintain the optimum conditions for the growth of microorganisms used in the large scale fermentation. So अगर आप एक you are using a apparatus to maintain the optimum conditions, to maintain temperature, pH, and various growth factors. So what is the characteristics of it? So it is a closed, vertical, cylindrical, stainless steel vessel. So as you can see here, this is a stainless steel vessel which is in cylindrical shape. Its size is from, it can take 5 to 5 million liters of liquid in it. So here I have drawn a picture of it. This is the motor attached to it. From nutrients are added from that side, a liquid is present in it. This is the water jacket which surround it, in which the cool water enter from here and it will remove from this side. So it is used to maintain the temperature of that fermenter. Here the temperature or pH monitor is also attached. The extraction of the process will take out by this side, and the nutrients are added from this side. Let's suppose these nutrients are added in this way. And this is the stirrer which is used for the mixing of all the contents, and from here the product is extracted out, and from here the sterilized air is also entered, which is used for maintaining all the processes in the optimum state. So this was all about the fermentation. So we have discussed what is a fermenter, what is the processes of it. In the next process we will discuss how the antibiotics drugs are prepared in this by the fermentation process. So this was all about the today's lecture. Hope so that you will do fine. And one thing you have to remember, that there are further two more types of fermentation, which is the alcoholic fermentation and lactic acid fermentation. So in the alcoholic fermentation, it produces alcohol and release carbon dioxide from the pyruvic acid, while in the lactic acid it produces lactic acid and does not release carbon dioxide from the pyruvic acid. Alcoholic fermentation take place in two steps, while the lactic acid fermentation take place in one step. So your alcoholic fermentation needs two enzymes, pyruvic decarboxylases, to remove carbon dioxide with the magnesium 2 plus, and your lactic acid fermentation only need one enzyme, which is the lactate dehydrogenase. And your alcoholic fermentation, it form acetaldehyde, while your lactic acid doesn't give an intermediate. ठीक है, लेकिन alcoholic fermentation has an intermediate which is the acetaldehyde. And alcoholic fermentation occur in yeast, while the lactic acid fermentation occur in the bacteria, some fungi and vertebrate muscles. So हमारे muscles में lactic acid fermentation हो रहा है. ठीक है. So this was all about the today's lecture. Meet you soon in the next lecture. Till up, Allah Hafiz.